Okay, so let's start today. So today is another one of the biological days. So we will start with some ideas about metabolomics. And metabolomics has to do with the metabolic system, so it has to do with the biochemical reactions that take place within the cell. So we have seen already a few of these labels. So we have the genome, transcriptome, proteome. So the DNA is translated into RNA, and RNA is translated into, this is transcribed, this is translated into proteins. And then these proteins are the ones that really perform the biochemical reactions within the cell. So the results, the products of those reactions. So the products of all these biochemical reactions are smaller molecules. So we can see sugars, nucleotides, amino acids, lipids. Here you have a graphical representation of that. So you have the DNA. DNA is transcribed into RNA. RNA is translated into proteins, and then proteins interact with each other and they produce metabolites. And you have an idea of the size of these molecules. So metabolites are really small things, and the number of these entities. So we have about, I don't care if it is 40,000, 20,000, 30,000. There are different estimates of the number of genes that we have. These genes are, they are translated into a larger number of transcripts, and then these transcripts, they are translated into about 100,000 million proteins, so somewhere in that order. And then metabolites, they're relatively few, so it is only about 3,000 different metabolites. And according to this. And then something also interesting is that this is more related to your nature. So what do you have in your genes? This is more related to the environmental influence. So which metabolites you have in the cell is partly determined by the environment. Okay, so yeah, some ideas of how this data is produced and analyzed. So you start with some sample, biological samples of blood, tissue, whatever. Then you go into an equipment that typically it is some kind of mass spectroscopy. These two are mass spectroscopy. This is NMR, so magnetic resonance. Then you have some spectra. And now you have to analyze this spectra. So there is a lot of low-level signal processing, and then at some point you have to analyze the results. And the part of the analysis will be identify the metabolites that are present there, and here which metabolites in particular is what you are looking for, and the databases, and you have to match the spectra that are deposited in these databases with the spectra that you have collected, and identify which ones here are represented there, and then you identify the metabolites that you have, and then you have to interpret those results. So what is the meaning? Why do you have those metabolites in this particular moment? And here is proteins are enzymes, and the enzymes act upon substrate. So the metabolites here are the substrates typically. And the idea is that the enzyme facilitates the interaction between the different metabolites, between the different compounds. So for instance, if these two compounds have to interact to form something else, if you allow them just to form this something else just by diffusion, it is a rather slow process. And then the enzyme puts both into an appropriate orientation, and this translates into a lower energy barrier, so that it is easier for these two entities to form a new product. So the enzyme lowers the energy barrier, and it does that. So enzymes, typically, they are proteins. There are also RNAs that can act as enzymes. And so you have a big structure, and this big structure in some little pocket, it is combining these two molecules to create something else. So for instance, it orients these two molecules in the right orientation so that they can interact more easily. So this is the way enzymes work. So now we are more focused here rather than in the protein. Okay, and yeah, and if two chemical entities are interacting, then yeah, this should be correct. This should be both ways. Okay, so it is a reversible reaction. So if X1 and X2 bind to form X3, then you can write the rate at which this reaction is happening. So you can put the derivative. So what is the rate of change of X1, the concentration of X1? So it is the same as the rate of change of the concentration of X2, and it is minus the one of X3. And all these three together, they are this one. So it is X1, X1 and X2 have to bind. So if you know already that this is the product of the two, there will be some constant. And because they bind and they disappear, there is this minus sign there. This is the mass action equation. Okay, and then let's say that we have a different. So this one would be represented here. Okay, so the constant is not the same one, but the idea is the same. So you have two entities and they combine into something else, both together. So let's say that you have a different reaction. So this reaction, two molecules of X1 that bind to form X3. So now the mass action equations are this one. So X1 decreases at this rate. So this rate is X1 binds to itself, so it is X1 squared. And then this is minus K3, and K3 is formed at this rate. So when X1, two X1s bind to form X3, then X3 appears exactly the opposite of this one. Okay, so this would be this reaction here. So no, this is not this one. This is this one. Okay, good. So you may have heard of this Michaelis-Menten equation for enzymatic reactions. So this Michaelis-Menten is coming from this same modeling that we have applied so far. So let's say that you have an enzyme, you have the substrate, and they can form a complex enzyme substrate. So it is kind of the first binding here. So in the first molecule binds the enzyme. So you have both together, and then the enzyme, let's say, that modifies the substrate and releases the product, and is released itself so that it can interact with another substrate molecule. So we can write the mass action equations for these reactions. So okay, so let's see how the substrate changes over time. So it binds the enzyme, so it disappears at this rate, and the enzyme and the substrate both together, they disassemble and they come back because this is reversible, and this happens at this rate. So S and E, they are the concentrations. So these are products of concentrations. This is the concentration of ES both together. Okay, now at which rate this one changes? Okay. Either it is formed, and it is formed at this rate, so these two are the same, or it disappears because it is translated into either it disassembles or it releases the product. So which is the rate at which those processes happen? So it releases the product with constant K2. Releases the product with constant K2, or it disassembles with rate K minus 1. And then the product is formed at this rate, changes at this rate. So it has to come from this one, and through K2, it is released. Okay, so these are the mass action equations for this reaction. So with these, you can simulate, or you can mimic what the concentrations will be over time. And then there will be a number of simplifications. We want to go to Michaelis-Menten. Michaelis-Menten is this one, okay? So there are a number of assumptions to go from these mass equations to Michaelis-Menten. So the first assumption is that we are at quasi-steady state. Okay, so there is much more substrate than enzyme. So we have a lot of substrate and very little enzyme. So that means that this one doesn't change too much. So if it doesn't change too much, then this is zero. So the rate at which this one changes is zero. And then these two terms have to be equal. And now you can rearrange. So if they are equal, you can take K1, put it to the right, ES, put it to the left. So this ratio, we will call it Km, and on the other side, what you have is this one, is this side. So it is just rearranging this equation when it doesn't change over time. There is another assumption, that is that this dissociation here, it is at equilibrium. So if it is at equilibrium, again, this change is zero. So these two terms are equal, and then you rearrange them again so that you can define this dissociation constant as the ratio of these two. And this is almost, you can look at this one, it looks very much like this one, except for K2. So, and then we can measure what is the occupancy of enzyme. So how much enzyme is bound to the substrate with respect to the total concentration of the enzyme. So here, the total concentration of the enzyme is the free enzyme plus the enzyme that is bound. But then this one, we know from here. So yes, you can substitute. So you can solve here for ES. The Km will appear. And now you can divide by E, and you have this thing. And we have the product. So the product, the rate at which it changes, it is K2 times this thing. But this thing, now you have it. You have ES is some constant time times the total enzyme concentration. So now you have it there. So now you have derived the Michaelis-Menten relationship. So the rate at which the product changes is this one, but this one comes only after a few assumptions. So if you want the raw equation defined in everything, forgetting about these assumptions, then you should go to these ones. So how is this related to system biology? So in system biology, we are trying to model mathematically the different processes that are taking place in the cell. So this one, now we know it is a simplification that sometimes is useful, of a more complex system that is actually the one that is taking place. Okay, so The Michaelis Menten curve is typically represented in this way. So this is the velocity of the reaction. So this is the change of P. So we can look at here. So this is exactly this equation here. So the rate, the change rate of P is called Gp, so the velocity of P. So the velocity of P is this thing. And if instead of looking in this way, you invert it, you compute the reciprocal. So now it is a line. It is a line, but in 1 over s. So this is a constant, this is another constant, and now you have the equation of a line. Okay, so all these things are familiar to you, right? But now you know the derivation of these things. Okay, so we can make this Michaelis Menten kinetics a little bit more complicated. So now instead of binding just a single substrate molecule, we can bind n substrate molecules. And then this enzyme here, this product together, this complex will have n substrates. And now the modification of the equation is I have put it in red. So it is the same equation that we had before, but now there are n's that appear in that equation. And now by making the same assumptions as before, now we come down all the way to this thing. So the n has appeared in these places. Okay, so this is called Hill kinetics. So it is a generalization of Michaelis Menten. And depending on the value of the n, so depending on the value of the n, you may have, well, this is Michaelis Menten. So for n equal 1 is Michaelis Menten. But if you have an n that is larger than 1 or smaller than 1, then you have larger or smaller slope at the beginning. At it's interesting. At this concentration, let's do it with this one that is simpler to see. So when the concentration of the substrate is Km, you are exactly at half the maximum velocity. So you can see this is Km, this is twice Km, so you are at half the maximum velocity. So that point is fixed. So that point also happens in this Hill kinetics. But the way that you reach to that point changes depending on what is the n. So you may go in a more steep way if n is larger than 1, or in a smoother way if n is smaller than 1. Okay, next. Enzymes have more complex interactions with metabolites. So they can have, in particular, we will see 3 of them. So they can compete with the substrate. So for instance, you may have an inhibitor that competes with the substrate. So the substrate should go there. But there is another molecule that also goes in the same place. And then the enzyme can go this path in which it is binding the substrate, or it can go this path where it is binding the inhibitor. And now you can make kind of similar reasoning to the one we did before. And now the velocity at which the product is R is our product. So the product is formed is this one. That is exactly the same as the Michaelis Menten, except for this term. So the larger the inhibition, the smaller the inhibition, so the Ki is a concentration, has units of concentration. So the smaller the inhibition, the smaller this value, the stronger will be the inhibition. Then the inhibitor can bind to a different place. So for instance, can be non-competitive. So here the inhibitor is competing for the same binding site. But here the inhibitor is binding somewhere else. And then someone, once it it is bound, it interferes with the binding of the substrate and releasing, especially with the releasing. So here we see the enzyme can go this way. That is the standard way. But if you add an inhibitor, even when it is free or when it is bound to the substrate, it forms a different complex and it stays there. So it binds the enzyme with the substrate and then it doesn't release it. So the product is never formed. And now this inhibitory term goes to there. Okay, so if you do the same kind of equations that we did before. And now a different way of working could be uncompetitive binding. So the inhibitor doesn't bind originally, but once the substrate binds, now the inhibitor can bind. And now this compound here, it is sequestered and it is formed something else, and the product is never released. And then the equation is this one. So depending on what you want to simulate, you may use one different equation. Okay, so there are databases of enzyme data. So you have, for instance, here, you have, this is a function. Okay, so this is a function. So you have list of possible enzymatic reactions. So you have transferases. Within the group of transferases, you have a set of enzymes that transfer phosphorus containing groups to the substrate. And then if you are transferring the phosphorus to a protein, then you are called a kinase. So this is the group of kinases that phosphorylate tyrosines and so on. So then, given one function, you will have different reactions here. So different enzymes that belong to this class. So you have, for instance, here you have ATP, you have this tyrosine, and then here you have 3 phosphates, and after the and in this reaction, one of the phosphate groups is lost from ATP and it is transferred to the tyrosine. So any enzyme that performs this reaction belongs to this group. And there are databases for that. And you also have different values for these for possible reactions that belong to this group. So this is the substrate, this is the concentration, and I don't know, this is a particular enzyme. I haven't put it here, but this enzyme on this substrate has this Km value. The same enzyme on this substrate has this Km value, so on. And where you have measured that, at which pH, at which temperature, and this is a part of a protein, so which protein is doing that? So this is the UniProt identity ID code of this enzyme here. And you have also Ki values. So this reaction is interfered by all these compounds. And here you have the Ki values of that interaction. Okay, and there are a lot of enzyme databases. So this is the list of classification of enzymes. This is Brenda is a database of enzymes, and there are many others. Any questions to me? Okay, so we have already seen that there is a lot of regulation in all these in all these biochemical reactions. So we have seen that the transcription of DNA into RNA, there is a lot of control there and all these transcription factors and so on. And then translating from RNA to protein, also there are a lot of regulation. Post-translational modifications change the way that protein behaves. So there is, again, a lot of regulation there. And here at this level now that we are today, the metabolism, there are also a lot of regulation. And this regulation comes from regulatory proteins. It comes from different pathways. So in the regulation of these, so you have to regulate the enzyme and you have to regulate the biochemical reaction at which speed this is happening. So there are possible ways to regulate these. And here I'm showing 3 of them. Genetic regulation we have already seen. Allosteric regulation is this one. So this is an allosteric. This one. These 2 are allosteric regulation. So the release of the substrate is regulated by a different binding site. So this different binding site is called allosteric. Then you can change the enzyme. So these reversible covalent modifications, you can break the enzyme. And there are a few, for instance, this reversible covalent modification. So you may phosphorylate. So sometimes the kinase is only active if it is phosphorylated. If it is not phosphorylated, then it doesn't work. So by regulating this phosphate here, you make the enzyme active or not. And feedback regulation. So let's say that you have a metabolite and then you have an enzyme. This enzyme transforms A into B, and then B is transformed into C, C into D. And this one has a negative feedback. So this one inhibits enzyme 1. So this kind of negative feedback, what do you think it will do? Negative feedback loops, they're always used to do something. To stabilize all these concentrations at fixed level. Okay, so if it, let's say that it increases. So let's say that D goes above the level that you like. So there is a lot of D, so there will be a lot of inhibition here. And if there is a lot of inhibition here, this D will be degraded eventually, and then the concentration will go down again. Sorry. So this is another way of regulating. And this is another way of regulating, that is isoenzymes. So isoenzymes, so for instance, this is a protein, lactate dehydrogenase, that is a tetramer. So it has 4 monomers that are bound into a single protein. But the different subunits have 2 forms. It has an H form and an M form. And depending on the tissue, you may have, so for instance, at the heart and erythrocytes, you have the form HHHH. So the tetramer is formed by 4 identical subunits. But in skeletal muscle and liver, you have MMMM. So the protein is almost the same, but with a small modification. So, and depending on which tissue you are, you have different composition of this enzyme. So these are called isoenzymes. And it is another way of regulating the metabolic levels of each one of the different compounds in each one of the tissues. And yeah, these are different techniques to measure metabolomic data. Most importantly, you have all these magnetic mass spectroscopy, many different versions. Any question up to here? Okay, so next, let's go to signaling systems. So signaling system is how does the cell know that there is something outside of it? So it goes through signals. So for instance, a small molecule, it can be an hormone, it can be a metabolite, it can be a small molecule that binds a receptor that is in the cell membrane. And then this binding is transduced into a number of chemical reactions here in the cell, inside the cells, and then it diffuses down to the nucleus, for instance, and the nucleus responds by activating some gene because it knows that there is something outside that is interesting or that it has to escape from or whatever. So, yeah, so here is the same idea, but now with a little bit more detail. So there are a number of receptors here that are the ones that are mostly involved in this kind of reactions, of detections. So GPCR is a whole family of molecules. Actually, most drugs, they affect the GPCR receptors. Then you have growth factors, you have integrins, you have cytokine receptors, and you have also these are antibodies that combine here some antigen. And yeah, and then once they bind, so the molecule binds the receptor, this molecule changes the conformation, and this change of conformation elicits a lot of reactions here, and you know already the convention. So for instance, these arrows means that this path is activated, while these not arrows, but these lines, red lines, this path is inactivated, is inhibited. So you can model all these networks with different tools. So for instance, you can use probabilistic Boolean networks. So these probabilistic Boolean networks, let's start with Boolean networks first. So Boolean network would say, okay, if the concentration of this molecule is high, then this is the receptor. This is the epidermal growth factor. It binds the epidermal growth factor receptor. So if this molecule is high, then this is negated. It is negatively affected by this binding. So these are inhibitors. And then, so if this is high, this will be, if this is high, this will be low. If this is low, this will be high. And then this high here causes a high there. So, and then it propagates and kind of circuit. And for instance, here in this paper, you have one of the pathways, this signaling pathways that has been modeled in that way. But you can also have probabilistic Boolean networks. So it is not that a high here always produces a low there, but it produces a low with a given probability. And then we saw this one of the first days, where you have here is the state of three molecules that you are interested in. So let's say this is one of the molecules is high, the other one is high, the last one is low. And then if this is your state, then you can go to this other state with probability 0.09. You can go to this one with probability 0.14. You may stay in the same state with probability 0.06. And you can model these signaling pathways. These signaling pathways here. You can also model either using this kind of mathematical models or using this kind of mathematical models. What else? You can also use Bayesian networks that we also saw one of the first days when we introduced all the mathematical models. And then here you have how these high and low concentrations in the different molecules, how they can change over time. So for instance, here you have how they can change over time, and then you simulate using these Bayesian networks. And the details of the Bayesian networks we saw on the first day. And going into one direction. Okay, and also you may use differential equations. So in differential equations, something that is important is the definition of stable states. And so, for instance, here, let's say that we have this differential equation. It is generic, so we are not modeling for the moment anything related to metabolites or anything. Just want to understand the properties of differential equations. So this says the rate of change of x depends as a third order polynomial. And so x1, x2, x3, there are three given values. Okay, so let's put them to -1, 0, and 1. And then let's say c is equal 1, and then this is the polynomial. So this is the rate of change of x. So you may remember from differential equations, if this is 0, if this polynomial is 0, then x will not change. Okay, so at x1, at x2, and x3, if you are exactly at x1, it will not change. So if it doesn't change, that means that this is a critical point, this is another critical point, and this one too. But these two critical points are stable, and x2 is unstable. So what is that? If, for instance, let's say I gave you this example at the first day. So this is let's describe the movement of this. This is an unstable critical point, while this one is stable. If I perturb this one, it tends to go to the same position. But this one, if I perturb it, it falls. Okay, so this is unstable. Why do I know that? Because of the second derivative. So the second derivative here. So the second derivative of x, that is the derivative of x. So this is the derivative of x. This is what we are saying. So the second derivative is the derivative of this. So here it is positive, here it is negative, here it is positive. So that means that these 2 points are critical and they are stable, but this one is unstable. So if you are at x equals 0 and you perturb it a bit, you will go either to 1 or you will go to -1. And if you are at 1 or -1 and you perturb it, this is time, and you perturb it, you will come back to the same minimum, to the same state. Okay. How can we model this with? So the kind of mass action equations that we are having, they introduce this kind of terms. So we have some production. So this is X is created, and here X is destroyed. So here in the creation, it is blue, so it is this curve, minus this one. So this one is this one, the one in red. So there are 3 possible stable points: this one, and this one. And if you analyze what is the second derivative, turns out that this one is stable, this one is unstable, this one is stable. So this is the subtraction of these 2. And here the derivative is negative, second derivative is negative, so this is stable. So here you have how it will behave with time. So let's say that you have a low concentration. So if you are at a low concentration, around 50, around 20 here, and you are perturbed, you go back. But if you are perturbed enough, then you may cross this barrier, and if you cross it enough, then you go to the other stable point. So if you cross it enough, then you go to the other stable point. And if you are perturbed again, come back to your stable point. And then if you perturb it enough, you may go down. So these are differential equations in general. What about system biology? So we are simulating the behavior of molecules inside the cell. So let's say that this is our circuit. So if this is our circuit, this is the creation. We look at this particular molecule, this T. So this T will behave like this thing. So this thing here says, okay, so the change of T, you are creating it, and then you are using it for something else, and then you are also destroying T. You have some elimination mechanism of T. So let's see. So this one is the creation pathway. So this one is modeling the creation pathway, and it looks a little bit like the mechanism, no? That's that kind of flavor. And then you have the inhibition, the degradation, and then you have another pathway of elimination. And you may draw these different lines. So this is the creation line, the blue one. The red one is the degradation one. And then there will be 3 stable points. So what we have here are B stables. So you have studied B stables in circuits, right? So here what you have is a B stable. So the concentration of this molecule is happy at 2 particular levels. So it is the same. That was happening here. So there are 2 concentrations at which it is happy. And if you perturb it around one of the happy states comes back. If you perturb it too much, goes to the other state. So it is a bit stable. And this is a very typical pattern. So in this typical pattern, so you have some signal from the outside. This signal from the outside affects Y. So this could be the receptor, for instance. So this receptor activates a different molecule X, and then the activation of this X somehow facilitates the binding of the substrate to the receptor. Okay, so let's see that. Yeah, and then there is some elimination here, and then some elimination there. Okay, so let's start with this one. So the substrate binds the Y. So substrate binds X to form Y. So substrate binds X to form Y. And then Y is eliminated. What about X? So the binding of Y promotes X. So it is in the creation pathway. And then there is a constant level here that is a constant creation. And then X is also eliminated. And let's put some constants. And now let's play with S. Okay, so let's say that S, we have a constant. So S is as a concentration of 3 all the time. So it is a some stable point here. So it has these changes have converged over time when you are at the stable level. And then suddenly S goes from 3 to 10. Okay, so let's see what happens with X and what happens with Y. So X and Y, if you simulate these 2 equations, they will rise, both of them, and they will reach their new stability level. So Y will go to here, X will go to here, and then they stabilize. They stabilize, and S now disappears again, So it goes down again from 10 to 3. So now you simulate, you see what the two concentrations of X and Y look like. And you would expect that, okay, it was 3, it was at some level, then it went up. And then if it goes back to 3, this should go down to the same level as before. But this is not what is happening. So what is happening is that it is getting trapped into some intermediate stable state. It is trapped there. And it doesn't fall to the original level until S goes down to 2.5. So when S goes down to 2.5, then it drops to the original level, and then it goes up to 3, and then you are at the same point as them. And this behavior, in which you are at some level, you go up, and then when you go down in the input, the state of the system doesn't go down to the same level as before. This is called hysteresis. Okay, so this hysteresis is somehow a memory of the system. The system knows that this 3 is different from this 3. And it is different because this 3 is after a high level of S, while this 3 is after a low level of S. And it has reached that conclusion through the stability points of this equation system. So this is rather surprising, I'd say. Any question after here? So just one more. No, we can leave it there. So let's make a break of 10 minutes. We come back at 5:30. So let's move on to the next step. So here is you will see these mechanisms. So let us see. For instance, you can take this one. Siempre tengo el problema este. ¿Sabéis cómo se soluciona o no? Bueno, si no lo dejo así chiquitito y ya está.
Con doble notación.
So this is a neutral field that is pursuing this point. So there is a chemical gradient. So this is this mechanism of a molecule that is outside the cell. It is binding some receptor, and then the receptor, this signal is transferred to the interior of the cell to change the cytoskeleton towards that direction. Okay, so another. So here we have how that is happening. This process in general is called chemotaxis. Después del anuncio. So for instance, this happens when a neutrophil has to go out from the blood vessels, and then what they do is they change the cytoskeleton. So this is a very long video, so you can change it. But it is the cytoskeleton that is changed, and then here it is degraded. These actin filaments, they are degraded, and then they are created in this direction, and then like that, the cell can move. Okay, so yeah, so here you will also see this is a different mechanism. So this is with with flagella. So these are some bacteria, and these are the flagellum of this bacteria. There are filaments like this, and there is a motor there. Then this motor is rotating. What is actually what is happening? So this is in the part. So let's go back to the beginning of the video. So here it is sensing that there is some chemical gradient that it likes. So it is looking for food, for instance. So there is a glucose gradient here. So it moves towards the source of food. So the signal is elicited here, but it is transferred all the way through the cell to the flagella at the end. And all here that is happening from the beginning to the end are these kind of biochemical reactions that we have seen today with these differential equations. And these differential equations at the end is a simplification. We saw it again the first day. It is a simplification of these energy landscapes that are, so when you have a molecule, you have an energy landscape of what are the different energy states that it can take. So what you are seeing here is just this energy landscape and differential equations in actions. So, and this is happening in reality inside the cell. So these are all these differential equations and energy states. Then because this moves, then the flagella moves. And and this is, if you want something to move, you need a source of energy. And it is this proton gradient, the source of energy for this motor. Okay, so yeah, you can see the videos yourself. So next, there are many systems, many organisms, archaea and bacteria in particular, that they use a two-component signaling system. Okay, so let's see what this is. So you have a sensor. Then this sensor receives a signal. This signal makes that this sensor phosphorylates. So this is the sensor, so the protein, the receptor or whatever, phosphorylated. And then the phosphorylated version of this one causes some response. So there is another molecule R that interacts with this one, but doesn't interact with this one. And then this one is also phosphorylated, and it causes this, or it propagates the signal down. And then this phosphate is removed from here. So the R takes the phosphate away from S. So the S is again available. So so this is a representation of that system, but this is another representation. It is an equivalent representation, but now with the arrows that indicate which ones are promoted and which ones are inhibited. So the signal promotes the phosphorylation. The phosphorylation promotes that R phosphorylates, and then these R that phosphorylate. So S causes this phosphorylation to come back, but then the signal can inhibit this branch. So this is a very typical circuit for detecting signals. Yeah, protein phosphorylation is this process in which you have a protein, and then you have ATP, and then there is a a couple of enzymes. One is a kinase. A kinase takes a phosphate from the ATP and puts it into the protein. And the phosphatase is the opposite. So the phosphatase takes the phosphate from the protein and releases. Okay. So this phosphorylation process. And many signaling pathways, they are regulated by phosphorylation. And so this was for mostly, doesn't mean that it is only. So archaea and bacteria, they tend to use this kind of circuits to sense what is the biochemical environment. And eukaryotes use a kind of similar one. So the process is also just phosphorylations, but instead of having 2 layers, it has 3 layers. And this layer in between is an amplification of this signal. So let's say that you have a very weak signal here, so very low concentration of anything here, and then this is amplified in this layer. And this amplification is made by these loops. So this kind of module is very typical in this signaling systems in eukaryotes. So where you have a protein, this protein can phosphorylate twice, and then once it is phosphorylated twice, it connects with the next layer. And here you can have hysteresis again. So if there is no hysteresis, you would expect that the behavior is like this. So you have some level. If the input increases, then at some point you will increase. But if the input decreases, you decrease following the same path. But if you have a hysteresis, then, for instance, you may go up following this path, but then when the level of the signal decreases, you don't decrease following the same path, but you wait a little bit more, and only after waiting you come back. So the signal has to really disappear quite a lot. So it has to go well down to elicit the inverse, or to come back to the rest state, if you want. If this is rest. Okay? So there are systems that behave like this, and there are systems that behave like this. So it depends on what is the specific enzymes and molecules that you are looking at. And this hysteresis is something that is very common in nature. So hysteresis happens when you apply a magnetic field to some magnet. It also happens when you change, for instance, the pH or the temperature of some environment. So you have an ecosystem, you change the pH a lot, or you change the temperature a lot, and then at some point you reach another stable point, and from this stable point to this one, it is not that if you lower the temperature, it goes immediately to this one. It goes through this cycle. And these processes, they are also involved, these signaling processes, they're also involved, for instance, in adaptation. So in adaptation, here is for instance, you may know when you have to prepare for some Olympic Games. So you know it will be very hot in these Olympic Games. So let's say that the Tokyo Olympics were particularly hot. So you have a couple of weeks of heat acclimation, and then that's some way to get used to these high temperatures, and then you can compete. And what you are changing is what are the levels, what are the stable levels in your in these concentrations that you are happy with. So which are the response? So you are changing this one. You are changing which are your states that are stable. And here you have a typical uh, circuit for adaptation. So let's say that you have a signal. This signal is at some, let's say, this temperature. So we are at some temperature, and then the temperature will go up, and then the response, you want it to change. So what it will do is at this level of signal, you will have this response. When it goes up, it will go up. But at some point, even if it stays, the signal stays, you don't want to be so high because this costs energy. So you want the response to come back, but you will come back to a different level. So how do you do that? So this is a circuit that comes back. So you have a signal. This signal activates A. A activates the transcription of B. So A promotes B and promotes C. So C is somehow the cause for the, so it is related to the response that you see. But then the activation of B inhibits the activation of A through the signal. So it doesn't matter if the signal it stays high because it is partly inhibited by B. So by constructing these circuits, you can adapt, for instance, and make sure that your rest level, your equilibrium state, they're different, and you adapt to different signal levels. Yeah, so circadian rhythms. So circadian rhythms is another different, it is a different kind of regulation that happens. So this regulation is related to something that we will see that are molecular clocks. So somehow it is that when there is a daylight, so we are day animals, so we like getting up early in the morning and then by night we go to sleep. So that is a whole, for instance, here, melatonin secretion starts. So how does the body know that it should secrete melatonin? But there must be some circuit that is making this cycle. Actually, the cycle is synchronized with the light. So if you stay very long without light, this cycle and the clock desynchronizes. For instance, this paper here, they look at a few molecules that are known to be related to these circadian clocks. And then this is the model that they propose. So there is a cyclin D1 that somehow there is a protein clock. This clock affects cyclin D1. The cyclin D1 has its own effects that are related to cell proliferation, cell differentiation. And then this one activates this path, but this path negatively affects this one. So there are, again, genetic circuits and biochemical pathways that are regulating these rhythms. And then at the core, what we need is somehow some kind of oscillation. So this kind of oscillation can be achieved with this system, for instance. So here what we have is that X binds S, and when they bind, they promote Y, and then Y promotes X, and Y is degraded and X is degraded. Okay, so we can put some equations to that. So for instance, Y promotes X. So we have this constant, sorry. So we have this constant creation of X, and then something that depends on Y. And then X is degraded, so X is degraded. Then X and S promotes Y. X and S promotes Y. and then Y is degraded. So, and then the signal may depend, and this is the key. So to make this an oscillator, we need that the signal depends on Y and X. And this is a model that is proposed, for instance. So S depends on Y, and then S is degraded. So if you analyze what is the response of Y as a function of S, this S is the signal. So if I increase it up or down, you see that the stable states of Y behave like this. And this is an hysteresis cycle. So if now this is we call this the phase plane, you remember? So you have you have a couple of variables, and then you see how you move along this phase plane. But we can also draw it over time. So we can see how S is changing over time and X and Y are changing over time. So you start at some level, and then you let those equations evolve, and this is what you get. So you have an oscillatory behavior in S, and this oscillatory behavior in S also causes an oscillation in Y and X. So now here you have the key for these circadian rhythm. So there are molecules that have somehow this kind of circuits, and then their evolution makes that it knows, depending on which are the constants, they can take a full day to start secreting melatonin again. And here is a very simplified version of this circadian rhythm. So this can also happen at the level of a protein. So for instance, you can have this protein. This protein has two phosphorylation sites. So there are two amino acids that can be phosphorylated. And then you have four states. So you have an unphosphorylated state, in a state S where only the serine has been phosphorylated. You have another state T where the threonine has been phosphorylated, and a state D where both are phosphorylated. And then you have this. So these four states are for a single protein. It is a single protein in different states. So actually they are kind of different proteins because of these states. And all these are reversible. And this is the regulation. So the S state activates another protein, Chi-B, and then this Chi-B inhibits Chi-A, and Chi-A activates this transition here, this transition here, this transition here. And so it activates this way back, but it inhibits this way down. And it has been proposed as a model for how these circadian rhythms can be held over time. You can see that the cycle is about 24 hours. So it depends on which are the constants of all these regulations. You can have faster or slower cycles. Homeostasis is another mechanism that is also regulated in this way. So for instance, let's say that you're doing exercise in the sun, so there is a lot of heat there, but our skin temperature is pretty stable. So you can see that the body temperature can go up very, not a lot, but it's 1 degree. But then somehow the whole system is regulated so that the skin stays very fixed. And the same happens here, for instance, with glucose. So this is the range of sugar level that the body likes. Below this one is bad for you. Above this one, it's also bad. So let's say that you eat something with sugar. So the sugar level in blood will go up, But then this is detected, and the cells that have that secrete insulin, they start secreting insulin, and then the sugar is internalized by the cells. So, and then the sugar goes away from the blood, and it is kept. And then when it goes to, because you're consuming this sugar, because you need it for your energy, then at some point it may go very low. This is detected. You go for something to eat, and the whole thing starts again. And all this regulation is regulated by the liver, pancreas, and some ways to communicate these two. And this way to communicate this glucagon, that is an hormone, and insulin. And here you have the same kind of circuits that we had before. So homeostasis is also, you can see it at the level of cell and genes and proteins, but you can also see it at the level of tissues and organs. And the same kind of equations, you can write the same kind of equations we have done for the genes. You can write it for these different concentrations. And yeah, and homeostasis is always related to some negative feedback. So there is a negative feedback here. So when something rises, at the end there will be some inhibition, so that when the level of the concentration of something goes up, it is downsized and it is decreased to the something is inhibited, and then you keep it. So negative feedback is always related to homeostasis. And there are databases of biochemical pathways. So there are a lot of them. Software to deal with this kind of stuff. That's it. Any question?